Where to start, Mario?
Ready, Anne? Whenever you want.
Hello and welcome everyone. This is our first webinar of the IUIS Junior Community Leadership Academy. We are very happy that you're joining us today. And my name is Anne Hahn. I'm currently chairing the IUIS Junior Community Committee together with Marion Brunk, who I will introduce in a second. And I wanted to quickly start with a brief overview about what the IUIS Junior Community is. So we basically want to promote and connect early and mid-career immunologists around the world at an international level, highlight the field of immunology, and also improve education and training environments. And if you want to hear more or follow us, we are on social media, Blue Sky and LinkedIn, and you can also sign up for our newsletter. So we are currently an umbrella organization for several other regional junior immunology associations, for example, the Young European Federation, as well as the Junior ALASI, which is for the Latin Americas. And we also recently launched the Junior FIMSA community, which is Asia and Oceania. And also the African Federation is currently establishing a junior community as well. So if you're interested in getting involved, here are sort of all the representatives of the individual regional federations that are representing you based on wherever you are, and you can reach out to them if you're interested in getting involved. So this is myself and Marion here, Adelaide for AAI in Canada, Sri and Misak for FIMSA, Sri and Eloana for YFS, Vasihun and Rotsiri for FICE, and Maria Julia and Isabella for Alasi. And I just wanted to also briefly highlight some of the activities we're doing. So this was us at the IUIS Congress last year, where we had several social events, but also a scientific symposium and a career fair. So this sort of is just an example of what we're trying to do. And obviously, we're here today for the Leadership Academy, which runs for the first time and is organized by Marion and I, with lots of support from different people, and especially the Mexican Society for Immunology, who is also hosting this webinar series, and where Marion will now introduce also the president for a few welcome words. And over to you, Marion.
Thank you so much, Anne, for the introduction, and I would like to thank the Mexican Society of Immunology and the IUIS for supporting those webinars. Before introducing the current president of the Mexican Society of Immunology, Dr. Eva Salinas Miralles. She's joining us from Universidad Autónoma de Aguascalientes in Mexico. Good morning, Eva. Thank you for joining us.
Good morning, Marion, and thank you. It's a great honor for the Mexican Society of Immunology to welcome you to the IUIS Leadership Academy webinar series. We are proud to host this space to strengthen our community. These seminars are meant to foster leadership in immunology. By working across borders, disciplines, and generations, the webinars will inspire new leaders who will guide immunology with excellence, inclusivity, and innovation. We are deeply grateful to the IUIS, its junior community, and to the organizer, Anne and Marion, for choosing our society to host these webinars. On behalf of the Mexican Society of Immunology, thank you for your presence and your participation. We hope these sessions will encourage fresh ideas and instill a deeper sense of responsibility for the future leaders in our scientific community. Enjoy this webinar series. Thank you.
Thank you so much, Eva. It's now my great pleasure to introduce our first speaker, Dr. Mirima Merad. She is currently Dean of Translational Research and Therapeutic Innovation at the Icahn School of Medicine at Mount Sinai in New York. She's an international leader in the field of immunology, and particularly renowned for her pioneering research on macrophages and their contributions to cancer and inflammation. Her laboratory now focuses on developing novel therapies and spearheads multiple initiatives that accelerate immunotherapy development, and especially fostering cross-disciplinary collaborations and translation of discovery. And I met Miriam in her role as president of the International Union of Immunological Societies, and she's currently the past president and was pivotal really in establishing the junior community and also gave the inspiration for this leadership academy. So it's our great honor that Miriam could join us today to share a few insights from her leadership journey with us. Thank you so much, Miriam, for being here.
Well, thank you very much, Anne and Marion. I remember when you guys decided to build this, initiative. I was inspired by your energy, and I'm delighted that you are carrying forward. I think fantastic to see people step up, and it's not always the case. And today I'm going to talk about that. I will try to talk about, Anne asked me to talk about my journey. I don't know whether my journey is inspiring at all, but I would just discuss mostly the anxiety and the doubt and the moments where you have to step up, hopefully, and if you relate to this, I just want to say that this field of ours is full of ups and downs, and we all share them. And this is what I want to emphasize today. So I'm of Algerian background. I was born in Paris just because my parents were studying there for a few years. So I was born in Paris. Often I am thought of being French, but I was born in Paris. I was raised in Algiers. My parents came back. I was raised in this beautiful city of Algiers, and just came back from Algiers. It's always a pleasure to go back, and I was raised in a family of physicians. I was very familiar with the clinical culture, and I became very interested in medicine very early on, mostly inspired by many members of my family. And I went to Algiers Medical School. I was there for 4 years. Unfortunately, there was a civil war at some point in Algiers that really it transformed the life of many others. And I left on around the 4th year, and I went back to Paris, where I decided to do a residency in hematology and oncology. I became interested in cancer during my medical school. I was very interested in a very serious disease. At that time, cancer was incurable, so many students were interested in oncology, and I decided to do a residency in hematology and oncology in France. And this is where really my interest in immunology came. I had never I was not so familiar with research. I was very much more familiar with clinical work. But during my residency, there were a lot of incurable disease, but there was the beginning of this curative treatment called bone marrow transplantation, or allogenic bone marrow transplant, was starting to cure especially kids with leukemia or very severe lymphoma. So it was the one curative disease for these very severe hematological malignancies. And what was very transformative at that time, and I followed it as a clinical trainee, is that while we thought initially that bone marrow transplant was curing cancer because it allowed to give high dose chemotherapy, there was this new data suggesting that the I don't know whether this is well done, because I think I wanted to show something else. Okay, sorry. I don't know whether you were seeing are you seeing my slides well? Even the end of my slides? And
Yeah.
do you?
Looks all good.
It's all good. Okay. At that time, there was this the realization that the anti-cancer effect was due to the donor immune system and not the high-dose chemotherapy. So there was a lot of excitement in trying to understand how the immune system can recognize and eliminate cancer cells. And at the same time, during my training, I was exposed also to many new type of successful clinical trials. There was the first success that cytokine could also, cytokine such as IL-2 mostly, and type 1 interferon can cure some subsets of very severe melanoma cancer lesions. And during an internship, not a rotation in pathology, I was realizing that tumor lesions were in fact full of immune cells, as if these immune cells were recognizing and eliminating cancer cells. So I decided to take a break and do a PhD in immunology. It was quite common that residents at the end of the residency could take, 1 to 3 years off to study a subject, scientific subjects of our choice. Many of my fellow of my peers were mostly studying cancer biology and really trying to identify new driver mutation. I became interested in these questions of how the immune system can recognize tumor cells and eliminate it. And at that time, there was a lot of excitement about developing cancer vaccines. So I decided to do a PhD in immunology. I obtained a fellowship from France, from the Arc Foundation, which in fact, France has been quite transformative for my career. So I'm very grateful for everything they've done, including funding that first fellowship, which really exposed me now to immunology, for the first time quite seriously. And I decided to do a PhD at Stanford. So at that time in France, you could have a PhD and do your research laboratory in different places. And I decided to move to the US, and I wanted to do a cancer vaccine using dendritic cells. Dendritic cells at that time were just discovered by Ralph Steinman. The idea is that, these dendritic cells would be injected in the skin of patients, pulsed with either tumor antigen, injected in the skin, injected, in patients, and that will mount an immune response against cancer and contribute to tumor elimination. Except that at that time, there was very little known about the origin of these dendritic cells. Ralph had discovered them 2 decades before. He was mostly describing their function. And the idea was that, and for this discovery, he received the Nobel Prize in 2011. Very side story, because he received the Nobel Prize, few hours or day after he died. So he never he never acknowledged, in fact, that the award. But at that time, when I moved to Stanford, the thought was that dendritic cells were derived from were cousins of macrophages, and similar to macrophages, they derive from circulating monocytes. And I wanted to mobilize monocytes to make more dendritic cells, so that, to expand, or as a form of enhancing the power of this vaccine. Okay, so during this journey, I said, let's learn about the origin of macrophage. And when I learned that, I will understand how dendritic cells come from. It turns out that these became, this took me to a very different path. I realized that these circulating monocytes were in fact not giving rise to macrophage in steady state. And then in fact, there was macrophages were self-renewing in different organs, and the monocyte-derived macrophages mostly in response to injury, but not in steady state condition. This was in contrast with the dogma at that time established by Van Firth and then Vincon that all macrophages were being constantly repopulated by a circulating pool of monocytes. So I decided that that was an important question. I discovered something that was quite new, and I have to say at that time, there was very little interest in innate immune cells. So dendritic cells were barely recognized as an entity. There was not so much respect for macrophages or neutrophil. The whole immunology field was focused on understanding, the development of, the TCR, the T cell repertoire, the B cell repertoire. They were cloning the TCR, cloning the BCR. So, the whole energy was around, this extraordinary cell type that convey immunology memory, and rightfully so, right? Like it was such an extraordinary, there was a lot of extraordinary discovery at that time. And here we were, me and several of my other colleagues, very interested in these cells. But I just thought that it was still quite important to continue to explore first that dogma was not totally true. Van Furth had suggested that monosaccharide give rise to macrophage. That was correct in the inflammatory setting, but not correct in steady state. And there was this hole that was not explored, and this is what I decided to explore. So I spent a lot of time then developing new tools, including this parabiosis tool that allows you to trace the circulating the progeny of circulating precursors. This is how I wanted to trace the progeny of this monocyte, try to understand whether they gave rise to macrophages or not in steady state. And I, instead of continuing my pursuit on vaccines, I really shifted my whole interest at Stanford on understanding the ontogeny of macrophages, thinking that I will come back to dendritic cell. I have to say the little trick there is that Langerhans cells, which are now recognized as epidermal macrophages, were thought to be the prototype dendritic cells. It turns out that these cells is a very interesting cells. I mean, I don't want to be technical here, so I'm being very high level in terms of immunology. I just want you to understand, some of the choices that are made that are not totally rational, but I think that choice that I made to move from vaccine to ontogeny was really was really driven by an intellectual pursuit, right? Pragmatically, it didn't make any sense in terms of my career in general, right? Like I was a physician, I was supposed to come back to France and lead a physician scientist program. I went there to make dendritic cell vaccines that can bring it back to patients. And here I was engaged into something that appeared totally esoteric for all my professors in France. And I have to say, I was often discouraged to pursue this path. I did it because the atmosphere at Stanford was so extraordinary and supportive, but that was, somehow a choice that was not totally not totally pragmatic. Anyway, so I spent some time trying to understand how this macrophage came from, and we realized there was a pool of macrophages that self-renew independently of adult hematopoietic precursors. And there I started to receive some offer to build a laboratory. And I decided to move to New York here. That was probably the most difficult decision of my life because I had to decide that I was posing clinical medicine. And in France, there was this rule, if you pose for more than 3 years, you'll have to, your ability to come back was going to be very difficult because you have only 3 to 4 years. I had a waiver already of 4 years to then do a clinical, what they call the clinica, which is the equivalent of, I don't know, maybe a fellowship here, not totally, but so I decided to pose medicine, okay, to start a lab in the US. And I was going to build my lab on 2 themes, continue these questions of ontogeny so that I could potentially build better dendritic cell vaccine. And then also I realized at that time there was a lot of macrophage in cancer, and those macrophages can also present antigen to T cells, so they could also potentially be harnessed to make a cancer vaccine. And so we spend time and trying to understand. So we define now this resident tissue macrophage lineage, which we become very excited about. There, I think here, somehow the community, it was not so. The community was very enthusiastic about the finding. we established there is a shift in our understanding of macrophage biology, that macrophage now are part, we describe them as part of as part of organs, the tissue cell, which program is tailored to each organ, and therefore they must have a very key role in organ health. And whereas this monosaccharide macrophage is really an effector cell type that come in response to an injury and has really an effector function. So we oppose these, and we become very interested, me and my small lab, in really opposing the role of these two cell types in health and disease. Okay, so we do quite well in terms of our ontogeny, but despite that, my colleagues were enthusiastic. In fact, we somehow managed to publish well, but my God, it was difficult to have grants. And I don't know whether there are many Americans here or junior faculty that are struggling to get their first grant, well, in the US or elsewhere, but I have to say it was very difficult for me to get my first grants. And I think for a few years in a row, I had everything that I had written mostly focused on these tissue ontogeny, and it was very basic ontogeny, was absolutely not well received by the NIH. So funding started to be very tight. I am unable to secure much funding for my ontogeny study. And I had these two big projects, and I said, okay, so now we have to regroup. No more cancer vaccine. We stop this cancer model. It's distracting us. We have to focus on what is most novel. And now I'm bringing the whole lab. Well, the whole lab was like 4 people or 5 people on this ontogeny, and we are all going to do that. And this is, and I think this was another big decision, which I still find today the most difficult part I find is to stop a project, right? When you engage, it's easier to engage in a question than to stop it. And and it was quite difficult because we had established some interesting new cancer model, etc. But so we had to regroup. And I have to say, in regrouping, this is, and focusing on one question, especially when you start, was quite, productive from us. So here, then things start to go well. We publish quite well. We managed to have grants, and then, okay, so after a few years, and I would say 7 years, things start to stabilize. And when things started to stabilize, this is where I went and I decided that, okay, so now it was time to really expand, take risk, and do different things, right? And this is where I decided that it was time to come back to cancer. And I will bring all these new knowledge to cancer. Now we had a distinguisher, and we went back to this field of cancer, I have to say, with some uniqueness, right? And this cancer field so when I came back, it was full of cancer immunologists, right? So much tougher to really, infiltrate or be recognized. But because we had that uniqueness, then people were very excited to invite us or to hear, and to hear from us. And the other things I've done is to move full force to human immunology. So we were having all these models, but it's very difficult to model chronicity in mice, right? So the anxiety that we had is that all these different macrophage programs that we were identifying in cancer lesions were maybe not so relevant. And I was, the lab wanted impact. Like, we wanted to make sure that this was real. So while we were designing and going back to models, we started a big endeavor to start to profile human lesions. And that was also a risk because again, the money was going to be tight, right? Like, again, we were going to diversify, and that strength of bringing all your resources into one question is often more successful. So diversification was a little bit difficult. So here I decided that I was not going to do this alone because it would have been too difficult. And I went to the dean of Sinal, and I lobbied to build a platform that was going to be distinct from my lab, that will allow us to really profile human lesions using the technology now. These technology are almost commodity, but 10 years ago when I started, I think we funded it around 2009. Single cell technology was just starting, and we felt this type of technology was very adapted now to be able to really profile with some granularity very small human biological specimen, and that will really help us inform all the program that we are seeing in mice, and really drive or allow us to design some reductional system where we will probe these hypotheses that will be made in human. And so we started, Sanji Kim, Schultz, and I, and now we are 30 people leading this platform, which is leading all these profiling. So now I have two different type of platform, and it didn't distract from my basic laboratory and all these human that all this human biology was being generated in a different platform. But this was not serving only my lab. It started to serve many different laboratories that Sana, my institution had invested in it. And now this is a very, very active platform. They look at thousands of samples, both blood and tissue per year, and we contribute to several consortia. So it's a very successful platform with fantastic people, several PhD. Many that we recruited from our program are now work there. But what I realized when I took over these platforms, everything was quite organic, right? It came from one question on wanting to understand whether this macrophage program were real, to building a platform and realizing this platform was not going to serve only cancer, but other type of program. And I realized that really this changed a little bit, because I will lobby or really encourage many of my colleagues to engage into human biology and using this platform, right? And I thought that we could do even more. We could now take this platform and potentially do the same thing in clinical trial. But I constantly had to convince everyone. And one day my dean's like, well, if you want to keep building things, that you have to take a leadership role. And I was a bit hesitant, because there's always a distraction when you take, the distraction away from the question you want to ask. But I was asked to take the leadership of the Immunology Institute, which is around 42 laboratories. It's mostly a scientific leadership, because well, anyway, I can explain how institute versus department works in the US, and I have to say I never regretted taking the leadership of this institute, because it became, I had spent a lot of time really thinking about some questions, being very granular, and it was somehow exciting to then export what I had learned in that one focus of mine to other questions and to other disease settings. So I'm now leading this Immunology Institute for more than 10 years, and it's been really a fantastic ride. So I'm going to go through quickly here, because I want to. So I built several. One thing I'm very proud of is really bringing different fields together. And this is maybe in science we do that much more than clinicians do. But in clinic, clinicians are trained to focus on one organ. And one thing I've tried to do is to really bring different clinicians together around themes, themes of immunology, for example, or theme of vascular biology. There are systems that really transcend every organ. So it was really bringing my scientific inquiries to medicine, and this is what this leadership allowed me to do. I'm going to go through this. One thing that we've done also through taking the leadership of the institute is it enabled us to bring things that we developed in the lab, not only my lab now. I'm talking about the being institute directors. I'm encouraging people to really bring what they discover and really bring it to the clinic. So we bring a clinical platform that really enable and facilitate this translation to basic scientists. And then if I want to finish with this one slide of lessons that I have for 20 years of being, an investigator, is that doubt and anxiety are part of the life of discovery. And this is what keeps us humble and keeps us creative. That it's very important, I believe, to pursue questions that excite you, because it's the only way to be transformative. If you are bored or if you do it because of career reason, I doubt that you can be very successful. I think it's very important to remain very granular on the question, but also have a high-level view on what you are able to address. Many times, including in at Sinal, I see scientists not having a sense of where the field is going. It's very, very, very important that you position what you are asking in the context of, the larger, in the context of your field. And it's like, what is your exact contribution vis-a-vis everything that's being done? I think it's very important to learn to pivot. I hope I described how we did it and how we went back. It's important to do it not too late, not too early. One thing I will encourage you to do is to be generous. I think it's very, very important to give to others, because our project is it's really a societal project. Nothing that we do it alone. It's impossible to do things alone. So it's very important to inspire people to join. It's important to show fun. It's important not to complain all the time about the lack of funding. Otherwise, people will not join our field and then we're doing something else. So, you guys, at your age, you are the one who can maintain this field alive. So please do it. Sorry. And then I wanted to, this is something I always say to those that can try to bring discovery to the clinic. If you don't do it, no one will. And it's your legacy of scientists to try to do this. You don't have to be the one to do it, but if you don't and you don't convince people to do it, no one will. And finally, there is no one single path to success. There are many different paths. I describe my path. I don't know, whether this is a successful path or not, but this is my path. There are many other very, very different paths that led to a lot of, successful science. So don't try to imitate, follow, but there are some principles that I'm trying to describe here. There is one thing I want to emphasize, because we are at a very challenging moment in society. And in the US, but not only, and many European countries are going through exactly the same problem, is that there is, I think, an anti-science movement that is spreading across the world. And it is our duty, I feel, to engage, and you don't have to engage and take the arms, but you have to engage in at least trying to describe in non-political terms, if you wish to, what you do to society. And we have to step out of the lab and at least take a little moment to educate, our family, our neighbors, friends in the school of our kids about what we do. Otherwise, I think we somehow have been margin of society because we trusted it. We trusted that we were, at a time in history where people realize that it is important that there are some scientists stuck in the lab and really trying to create new knowledge. This is not the case anymore, right? We have to go back and earn the trust and really explain, the pursuit that we have and regain the trust. We lost it during COVID, where somehow we lost the opportunity to win the public narrative, and we have to understand why. And this is the time to engage. And I just want to point, I just wrote this article on describing that what we do is as important, the academic research is as important as military. We are here to defend society. And I have to say, I have several of my friends, non-scientist friends, have read it. And for the first time, they said, true, that maybe, somehow we are as important as other institutions in this country. So please engage if you can, and I'm going to stop here. I hope I didn't take so much.
Miriam, thank you so much. This is exactly what we had in mind, and we knew you were going to expose so well about why what we are doing is important, and give us some advice to go into the future. Thank you, Miriam. We'll move on to our next speaker, who is Yamine Belkaid, and it is my great honor to introduce Professor Yamine Belkaid. She's the president of Instituto Pasteur since 2024 and also a world-leading immunologist as Miriam. Her pioneering work has transformed our understanding on how the microbiota and immune system interact to maintain health. Before joining Pasteur, Yamine built an extraordinary career at the NIH and has received multiple international recognitions. Beyond a scientific career, Yamine and Miriam really both strongly believe that investing in science is vital for society. Yamine has said that the true value of a scientific career lies in the people we accompany along the way, and she demonstrates that again today by joining us to share her leadership journey. On a personal level, I've had the privilege to meet Yamine on various occasions, and I am deeply inspired by her intellectual acuity, poise, and kindness. Yamine, the floor is yours. We are truly honored to have you with us today.
Thank you so much. I seem to have an issue with sharing my slides, but I'm going to try again. And if I'm not, I will just don't know what's happening. There is something with the sharing slide that is forbidden. Okay. Okay.
We go with the flow.
Well, you know what? We're going to go slide 3. I think Miriam had actually wonderful one before, and I'm just going to go. So I think there is a lot of commonality, and there's a lot of things that Miriam said that resonates with me. I mean, a lot of what she has actually shared: generosity, engagement, anxiety, but just going for it. I mean, all these remarkable values that actually I think are extremely important building a scientific career. And I think very much like Miriam, I was also originating from Algeria. I was born in Algeria, so that's a slight difference, and I was raised in Algeria. I became a biochemist in Algeria, at the University of Pasteur. And this is in Algeria that I really discovered my passion for science and really for infectious disease. I was very privileged when I was in Algeria to do internship at the Pasteur Institute of Algiers. I was starting to work on, of course, at my own level as an undergraduate on developing new diagnostic tools for leishmaniasis or schistosomiasis, with this big hope of developing new therapeutic tools. But this actually gave me many insights in what will become my journey. First, I was hooked. I was absolutely hooked by actually biological research. I was hooked also by science that connected with disease. And I was also hooked by field work, which in fact is something I haven't gone back to, but something that I really found extremely inspiring. And then this is actually where I discover also my one question. I think Miriam has actually described her question, but I think one thing we don't talk enough is very often the question that will drive you is the one you have identified very early in your career. This question is going to change shape, it's going to change expression, but there is one question. And for me, that was, how do microbes persist? For me, that was the most extraordinary challenge. Here we are, individuals and immune system, and somehow a microbe is able to actually sit in our skin, sit in our lung, as in the context of tuberculosis, for example, Mycobacterium tuberculosis, for years and years, and we are not capable to reject them. So this question really became an obsession. I mean, it became like, how is it possible? And this really what drove my entire career and actually drove and opened new different areas. The first one was really to try to continue this journey, and I did my PhD in Paris. At the time, it's a moment that we shared, of course, with Miriam. Algeria was going through extremely complex time. We had a civil war, and I wanted to stay in Algeria. I mean, I had no intention to move. I wanted to actually develop a scientific career in Algeria, but the graduate school closed, and I went to France. Again, Pasteur Institute, as you can see, there's a bit of a common theme there. And in Pasteur Institute, I started to do my PhD on very much the question I have mentioned. How does the immune system allow a microbe to persist? I didn't go very far. This was a PhD, big ambition, little tools. But this was nonetheless something that really, once again, brought me in the direction of those questions. Post my PhD, I think we talk about doubt, we talk about incertitude. I was convinced I was incapable to be a scientist. I had enormous doubts about my capacity to actually just develop a career. And in fact, I had no real clear idea of what I was going to do. And one of the questions you guys ask us is actually tell about a moment, something happened that really defined your journey. I think, frankly, I can take all the big decisions later, but the one that had the most impact on my life was this really bizarre moment where I was completely lost, where I was a single mother, and where I decided to go in the US in a laboratory from someone I never met before to work on leishmaniasis, and where I had very little control over the future or the comprehension of what was going to happen to me, not speaking the language, and sometimes jumping. I think these moments where we jump in a bit of the unknown perfectly, to be perfectly honest, and all the positive that come out of it. How you grow out of this experience, how suddenly new opportunity arrive. I think this moment really was defining for me, and actually that's how often I took those decisions. Was actually based on actually this first decision I took that really led to this path of not being in control, not be very sure of myself, but still giving a try, realizing that the worst thing that could happen is not trying. So I think this was, to me, you talk about the moment, frankly, this moment where I was like this 26-year-old lost graduate student is probably what has been the most important for me in terms of defining my career. So then I arrived at the NIH and I arrived in NIAID, which was an institute led at the time by Tony Fauci, which is a hero of mine. Tony Fauci, which many of you that are in this audience today live extremely difficult time as a courageous scientist, as a scientist, public servant that actually fought for what he believed, fought for actually the common good. And his vision of actually just science and research on infectious disease at the service of society was very much a big inspiration for me. And he did that not only by promoting science within the US, but in fact, by doing actually extraordinary things, like, for example, leading the development of the PEPFAR program, which many of you have heard, which saved thousands and thousands of human beings. And it's actually the disintegration of the PEPFAR program is going to actually condemn people. So this was a very brave scientist that actually put himself at the service of society, of HIV first, and then actually other infectious disease that really inspired me enormously. And in fact, had a very big role in my career subsequently. And then I was continuing with my little question, how microbe persist. And then I was really lucky enough, and this is actually where come, serendipity. I'm here working on leishmaniasis, trying to understand why those microbes persist in the skin of animals. And then I'm a little bit lost in my project. I see all these lymphocytes in the tissue, and I decided to go to see a talk at the time by someone that was working on regulatory T cells and working on actually regulatory T cells in the context of autoimmune disorder, something that had nothing to do with actually what I was doing. And this also changed my career. Just going to see this talk outside of my zone of work, my work was not very much strong in immunology, embracing actually another field and bringing back to my questions, realizing through this knowledge that actually regulatory T cells, a cells important, of course, for suppression, was required for microbial persistence, was really the one discovery that during my postdoc, really, as very much Miriam described, you need one discovery that is actually becoming, the beginning of your career. This was my discovery, and it came really by a mixture of obsession about my question, but also exploring how other fields could somehow provide an answer. And this is really the advice I keep giving to anyone I know. Do not stay within the constraint of your field. Look out there, look in neuroscience, look in actually physiology, look in medicine, being inspired by things that may seem off, but if you really have an open mind, they will feed your question in a way that could have extraordinary outcome. So following that, finished my postdoc, once again, completely lost. I have absolutely no idea what to do with my life. I had published, I think I had a good postdoc, but I had absolutely no confidence in my ability to develop an independent career. So I took a staff scientist position at the time. And nobody around me really looked like me. I mean, I was a woman, I had children, I was surrounded by guys around. I came from a different country, so I didn't have all the codes. I may not understood exactly what meant actually the codes of actually navigating a career in an environment that is different. So I really didn't have any, and I didn't have people around me that really told me that I could really go forward. And this really completely by luck that I met Steve Katz, that actually was a director of an institute that really at one point saw one of my talk and said, you should absolutely go for it. And from this lesson, I have learned one thing very important. When you meet people that have talent, tell them they have talent. When you meet people you believe in, go with them. Just be a proactive mentor to anyone you meet that you believe in. And I think, I know Miriam is someone that has done that enormously. Has been incredibly encouraged to training. We have to be proactive with the people we believe in. Do not actually allow them to wander around. Do not allow career to fall to the crack of maybe meeting someone or not. Be there. Be a mentor of people you believe in. So this actually was lesson one. It's good to actually have mentor. Lesson two, maybe I could have asked for help. I was there completely lost, not asking questions and actually waiting for someone to give me an advice. That's not the way you should conduct your life. I think your life should be also asking for help, not being actually afraid of asking for help. That was the second lesson I have learned. I should have been more proactive. And this is something I also encourage my trainees to do. I'm here for helping you, but go to grab help others. Be proactive. Be an agent of your own career. Do not passively wait for things to happen. They will not happen. Just go and take charge. Do not be afraid to ask for help when you're lost. Do not be afraid to be vulnerable. Do not be afraid to actually go with people that inspire you for advice. So I started my own lab at Cincinnati Children's Hospital first, realized and actually, maybe I can do this job. This is something I can function, continuing with my questions of how microbe persist, how regulatory T cells function. And then I started to realize that I wanted to actually take a turn. We talked, Miriam actually talked about the ability to do turn, extremely important. Very important to be focused early in your career. I think that's absolutely true. You have to be sharp focused. You have to be the best person possible in the question you have asked. But be careful to not become locked by your question. Because the day you actually become, you can actually, and I have seen actually people that were extraordinary at the beginning, but they stay so focused without the ability to turn, then one day they became obsolete. Your ability to make those turns is going to define your career. And these times are terrifying. Every time you do it, you are convinced this is the end of the world and you're never going to succeed again. But these are defining. So don't be afraid to be a bit lost. The moment of confusion, the moment when you feel like your lab is going in a direction that makes no sense, are in fact a moment of creativity. They are a moment of resettling. They are a moment, it's going to take time, and everything that is worth to do in life will take time. So just those turns are very important to do. So I wanted at the time to actually go back to my original question, which is microbial persistence, and the ultimate persistence is the microbiota. And this really led me to actually begin to embrace, before this became more trendy, the role of the microbiota in the immune system, physiology, regulation, and many others. But this also opened new doors because the microbiota interface with the immune system. I continued to explore the wonders of the immune system. I mean, Miriam talked about this revolution around the immune system and therapy. Well, I think this came because all of us started to realize the immune system is so much more interesting than just a weapon. It is a tool for regeneration. It is a tool for actually cognition. It is a tool for actually physiology. It's even a tool for the transformation of the body of the mother during pregnancy. And those questions really became absolutely fascinating to me. It's beyond host microbe interaction, how the immune system is an infinite source of manipulation of the physiology, and how we could tap into this knowledge for therapy, for regenerations, for longevity, for decreasing the inflammation. I think the immune system is one of the most extraordinary elements. It's actually a real star of the whole physiology. And I think the wonders of the immune system and the therapeutic opening of the immune system are just at the beginning. I think this is going to become even more important for anything we do. The immune system touches all physiological systems. So this became really inspiring. And then along the way, I had another privilege, which is to be joined in my journey by fantastic people, remarkable trainees, some of them that are actually now leading laboratory across the planet. And really more and more, by mentoring people, by creating community, very much like Miriam has done, I realized how much actually I enjoyed actually empowering the next generation of scientists. But this also gave me a sense of responsibility. What is the science of tomorrow? What is the community of science of tomorrow if we're not here also to protect it? And this really led me to another jump in the in the void when I left the NIH pre-Trump to join Pasteur Institute as the head of Pasteur Institute. This was again a really quite a strong turn in my career. You know, I was comfortable at the time. It was still comfortable to do science at the NIH. Things were going well, the laboratory was going well, and I really left at a bizarre time. Everything was going fine. I mean, there was no reasons for me to make this turn. But this turn was about what means actually a career if we don't really try to have the maximum impact on what we believe in. And what I believe in is science. I'm really obsessed with what science means for society, what it means for all of us. But I'm also obsessed about how protecting the next generation of scientists. How are we going to create an environment that is actually generous enough, that is collaborative enough to make sure that actually science remains empowered, not only visible, not only here to protect values, protecting society, but also that we let the space for the next generation to actually thrive. So I took this leadership position at Pasteur Institute because for me, it was one of the places that correspond to a lot of the value I wanted to protect. Science, a science that is connected internationally, because a lot of Pasteur I connected to Institut Pasteur across the world in a country that are LMICs, because also this is an environment of training, because it's an environment of advocacy. In many ways, this was also not only a mission, but also a platform to actually just be here, to be here as a guardian at a moment where, as Miriam mentioned, more than ever, science needs to be protected. So all of that is to telling you that I think I'm very inspired by Miriam, that is actually a dear friend and actually an incredible scientist. But I think there are a lot of people that are really right now have those strong value in science. I love actually to see the next generation growing because I really think the next generation sees science in a way that is so less personal and so much more at the community level. I think with the next generation arriving, we're going to build a science differently. We're going to build a science more collaborative, in which the sense of ego and self is less here, but the project and actually the mission is actually more represented. So I'm really looking forward to see you guys here taking the lead, honestly. I think my job is, as others, to hold the fort until you guys can take power. So that's it.
Yasmine, thank you so much. This is so inspiring. It's very rare to hear both of you really talking about your personal takes on your careers and your impressions, and I feel very privileged to witness this today. Thank you. So we're gonna move on to our quick Q&A session. We've got some questions in the YouTube channel. So I think there is one first from Miriam. Thank you for the great talk and sharing your personal experience. What were the most challenging aspects of getting engaged in science and oncology research at the time when you faced the decision, the decision to pivot?
The decision to give up. Can you?
What were the most challenging aspects of getting engaged in science and oncology research at the time when you faced the decision?
Yeah, well, first, it's interesting because I also resonated what Yasmine said, is that the naivety when you decide to jump, right? Like, so here I discover, this field of immunology at that time. It was not at all, it was the pre-checkpoint era. It was like this obscure group of people who were going to not target the cancer cells, but try to harness that immune system. It was, a departure, right? And so there was anxiety of engaging into something that was unconditional. Then the big decision was to stop medicine. So here I was trained for 12 years, and not being able to practice. So, it's a big decision. And then the decision to let a little bit that cancer immunology focus, which was at least made sense of everything that they've been, and go into very simple, very granular questions on trying to understand the ontogeny of the cell. So each time yes, it was scary, and also, it is scary, and also you are alone, facing this alone. So you can share it, right, with others, but there is something deep down that probably tells you to do it, because otherwise you would not have done it. I remember discussing this with my mother, and I said, "Me, why am I doing this?" She said, "You know what? No one forced you to do it. If you are doing it, is that something is that," and those instincts, I think, are very scary, but I would say take them, like if you feel strongly about something, but of course, because every decision, I have many trainees now, and sometimes when I say this is crazy, there is some crazy one. So you have to, you have to think about it and engage intellectually. And if it makes sense after, and well, then go for it. So it's very difficult to tell people to take risks, right? It is very difficult to teach someone to take risk. I do it. I have two sons, and you have to take the risks that are exciting for you, but you still have to have a little path, not everything designed, like everything mapped out, but somehow it has to make a little bit of sense. And then jump, because it is exciting to take that risk. And it is true that the statu quo, especially for us, I think, if we engage in this field, we probably like, that discovery sense. It excites us, and each turn and new turn that you take provide that level of intellectual excitement that I think is a driver for everything that we do. Well, I don't know whether it's a good answer. It's very difficult to discuss about.
Sure. Thank you, Miriam. Adeleye is here in the chat with us. Go for it, Adeleye. You can switch on your microphone. Yes.
All right, thank you very much, Mario. Yeah, I want to appreciate both speakers, both Miriam and Jasmine. So I have a question for Yasmin. That's about the fact that you mentioned that being at NIH as a staff scientist at some point before you were able to pivot into PI position at Cincinnati. I mean, I used to think that it's quite challenging when you take up a staff scientist position, especially based on the funding landscape or the fact that based on timeline as a postdoc, you need to transition and the faculty position is not coming yet. So how easy was it for you then to switch from a staff scientist position to an independent position? I don't know if you can comment or give advice on how one can make that a viable option.
Well, I mean, I think for me, the transition was more about having no confidence in my ability to become independent, to developing this little noise, like Miriam talked about, this little voice behind that begin to grow, and starting to hear to it and realize the malaise I was in this position. I was not comfortable. I was feeling that I was not breathing, that I needed to actually express myself. I needed to move forward. And so for me, I had to move. I mean, it was not, and then suddenly meeting this person that tell me you can go was just the little thing I needed to actually just have the extra courage to do it. But it was not easy because those positions are stable position. When you finish your postdoc, the first thing you want is a job. In many circumstances, you have 2 young kids. You just feel, okay, I'm going to get a job. And so it's jumping. And, getting grants is scary. I mean, am I going to be able to survive? Is scary. But at the same time, you do this job because you are passionate. And you do this job because you are actually an independent thinker and you actually an adventurer. I mean, I always find that I think there's an analogy. I know that we seem kind of a tame adventurer because we're in our office or laboratory, but we cross oceans. We have in the scientists, there's the same drive than the people that have crossed ocean. This absolute necessity to understand, to go to the unknown. And I think when you have this drive in you, and if you suppress it too long, something dies in you. So listen to it. And I think, when you have this absolute desire to express yourself, ideas you want to actually push forward, really listen to it. And if it's not the right environment, try to change the environment until you can actually just develop these things, and choose an environment for your training or for your mentorship around that is conducive to that. Don't suppress this discovery, this adventure energy that you have in yourself.
Awesome. Thank you so much. I'm trying to summarize two questions we got, which is for both of you actually. What do you think is the most important quality for a young scientist who wants to become a good leader? And also thinking about becoming a leader at the time you had to versus now. Do you think it's easier or harder in the current, let's say, political and general situation? That's a hard one, I guess.
Miriam, you want to
Miriam?
start this one, or you starting this one and I continue?
Well, I would say, well, leadership. Well, first, I think before becoming leader, you have to have something to say. So make sure you have, a vision first, and then leadership quality. I think to me is generosity, is really giving back and elevating others. So I would say these two quality, excellence and generosity.
Yeah. I mean, I resonate completely with what he said. I mean, I think there is one point you have mentioned, which I think is true. Resilience is part of it. Generosity is an absolute necessity. I mean, I think it's true in science. And I agree that actually before it's not becoming leader, first you need to become an expert at your craft. You have to be the best scientist you can be. And you have to be the most unique scientist you can be. When you have found this unique voice, people will hear. But don't try to be a leader, or don't try to actually just go if this is not how it works. It works because you are an expert and people respect you. Science leadership, in all leadership, but in particular in science, can only be achieved if people respect you scientifically. And then the second thing, is it harder?
I would say
Yeah, it
something
is
about
harder.
Yasmine when she went back to Pasteur. Just an anecdote. Pasteur is a tough place, very tough. And people were saying, wow, Yasmine, how people are going to listen. French never listen. But, there was, I don't know whether there are French scientists here. But I think that one thing, Yasmine, I have to say is that people listen because they respected your science. I respected you as a scientist. And this is how she rallied. And I think you have rallied so well, this group of scientists, which I think it suggests that, yes, the first thing to be is to inspire with who you are. And then, of course, there are all this extra quality that Yasmine have plenty of. But it's interesting to, think about that time where you transitioned. Yeah.
Yeah. So, I mean, one of the question was actually, is it harder today? Of course, it is harder today. I mean, the world is harder today. And I think this is come back to the point that is mentioned. We have to be generous. We who are at the stage of our career have to be generous, in particular today, because it is, of course, harder. I mean, I think it seems that at the time, maybe I didn't know where I wanted to go, but it was easier to have a career. Let's face it. Paper. I mean, I remember the first paper I wrote, it was like, it was like one ELISA per panel. I mean, let's face it, today it's like a multi-consortium of like 20 different. Yeah, so the reality, it is harder. But at the same time, we're learning how to do science differently. And I think we collectively have to be more generous to make sure that we give a lot of space to the next generation coming.
Thank you both. Miriam, did you want to?
No, I just want to say it's a tough time, guys, but we can't give up, right? We can't keep saying, it's tough. We have to just inspire others, because these kids otherwise are going to do something else. And there's nothing worse than, not attracting those bright minds to our lab, right? So it's our job, you guys, our jobs to make sure we keep recruiting talents. But and when you see, young kids, this is what you have to continue to inspire them now more than ever. So yes, it is tough, but there are also many extraordinary opportunities. This technology is changing, the way we are learning now about biological processes. AI is going to accelerate discovery. So it is an extraordinary time. When you look at either during our adult life, Yasmine and I, we have seen so many therapeutics being developed and curing disease. It's extraordinary. Like TNF blockade is my adult life, IL-17 blockade, checkpoint blockade. It's extraordinary. This is what we have to convey. So I want to finish with optimism here, because if we are not optimistic, our field is not going to survive. Yes, we are going through a crazy time. Me here in the US, I have to say, every day I wonder how is it possible that this government is still in place, one more day. But yet the community around me is the same, and we can survive this. There are other times in the history of humanity where there was this axis of evil that we are seeing now. But we have to remain strong, and I would say, there's something that's going to happen to our field. Maybe during COVID, we saw it, is that we are going to bond together again. We are going to partner again, because we realize that we need each other. So we may came out stronger because of all the craziness and the pushback that we are seeing.
Yeah, I fully agree with Miriam, and I think the last point I wanted to say is, yes, things are harder. It is true, and I think it's true for all of us, and it's true especially for the next generation. But I have yet to see someone with true passion that hasn't succeeded, really honestly. I mean, everyone I have met that is really, really driven, they succeed. It's never happened to me. So the people that have this light behind the eyes, this drive to actually just discover, that it always end up working. So clearly the magic is still there, and our ability to actually just push the boundaries is still there. And actually I have, as I said, in my entire career, yet to see someone with true passion and the quality we described that hasn't actually succeeded where they wanted to be. So I think that's, to me, the best hope for the future. And it's absolutely there. The space is still there for the dreamers and the adventurers and the people that will actually push the boundaries.
Yeah.
Thank you so much. I think out of respect for Yasmine and Miriam's time, we have to stop here today. So I'd like to thank you for joining us today and for making the first webinar of our Leadership Academic series such a wonderful start. Again, thank you so much for both speakers. It was so inspiring, and I think a very rare opportunity to listen to you in this way. So this is just the beginning. On September 10th, please join us again at 10 a.m. time of New York, 8 a.m. time of Mexico, same time as today, for exploring new horizons, pursuing research away from home with two extraordinary Brazilian immunologists who are currently building remarkable careers in the US, Camila Coelho and Carolina Lucas. We hope to see you there. Thank you again, and see you very soon.
Bye-bye.
Good. I think it stopped streaming. Thank you so much. This was wonderful. Thank you, everyone who joined.